Science, technology, and society is an interdisciplinary field that studies the conditions under which the production, distribution, and utilization of scientific knowledge and technological systems occur. The consequences of these activities upon different groups of people, science, technology, and society builds on the history and philosophy of science and technology. In Chapter 1, I will discuss the relationship between science and technology, the nature of science, the biology, and century. What is science and what is technology? Science is a way of understanding ourselves and the physical world. Process of asking questions and finding answers, then creating broad generalizations. Technology, the totality of means employed to provide objects necessary for human sustenance and comfort. The nature of science. Science is a process, not a list of discoveries. Science can prove a generalization not true, but cannot prove that is true. Biotechnology, the use of living organisms or life processes to solve problems or make useful products. Modern biotechnology, technology using cells and biological molecules or cellular and biomolecular processes, specificity, precision, and predictability, unity and flexibility, reproduction and renewable resources. Types and applications of biotechnology. Biotechnology and its industry. Biotechnology, bioprocessing, cell culture, recombinant DNA, monoclonal antibody, biosensor, microRNA, protein engineering. Industry, human healthcare, agricultural production, food and beverages, enzyme industry, chemical manufacturing, energy, waste treatment. The history of crop genetic modification. Stage 1. Genetic modification through seed selection. Stage 2. Genetic modification through plant breeding and selection. Models of inheritance. Fluid blending model. Mendel's theory of inheritance. Stage 3. Science-based plant breeding. Stage 4. Discovery of the nature of genetic inheritance. Plant genetic engineering. Who is Mendel? Gregor Johann Mendel is known as the father of genetics. Mendel coined the terms recessive and dominant. What is recessive and dominant traits?
Traits in humans can be either dominant or recessive. A dominant trait masks or covers up a recessive trait, and a recessive trait only appears if you inherit two recessive alleles. As you go through this list of dominant traits, count how many you have. Let's begin with a low resting heart rate is dominant from a high resting heart rate. A widow's peak is dominant over a straight hairline. Facial dimples is dominant over no facial dimples. Your hair going in a clockwise direction, left to right, is dominant over counterclockwise hair. A cleft chin is dominant over a smooth chin. An extra finger or toe is dominant over normal. A straight thumb, dominant over a hitchhiker's thumb. Freckles are dominant over no freckles. Short fingers is dominant over normal-sized fingers. Believe it or not, webbed fingers, dominant over normal. And finally, a Roman nose is dominant over a less prominent nose with a bridge.
What is genotype and phenotype?
We're going to talk about genotypes versus phenotypes. So when you have a trait, that is a pair of alleles, such as big B, little b, or little b, we can refer to them in two different ways. The first is the genotype, which refers to the exact pairing of the alleles. For example, if we're dealing with eye color and there are two alleles, a big B for blue eyes and a little b for green eyes, then there are three possible genotypes: big B, little b, or little b. Right? So these are the genotypes. These are the possible pairing of the alleles. The genotype isn't concerned with the expression of the trait, only the pairing of the alleles. Okay. The other way to refer to a trait is by the phenotype, which is its expression. So using our previous example of eye color, there are only two phenotypes: blue eyes or green eyes. Right? The phenotype isn't concerned with the pairing of the alleles. Right? The genotypes big B, and big B, little b both give the same phenotype, blue eyes. Okay, so the phenotype is just the expression. All right, so to recap, the genotype refers to the pair of alleles for a trait, in this case, the specific letters, and the phenotype refers to the expression of that trait, right? How it looks or how it functions.
The nature of genetic material. DNA as genetic material. What is DNA? DNA stands for deoxyribonucleic acid. It can be found in the nucleus of the cell.
Developed two fundamental laws of genetics. These are the law of segregation and the law of independent assortment. To see how these laws were derived, let's examine one of his experiments. Let's say purple flowering and white flowering plants are hybridized. We would find that the F1 generation is entirely purple. No white, no pink, just all purple. This experiment alone proves that any kind of blending hypothesis must be false. But even more perplexing is that if the F1 generation is allowed to self-pollinate, the F2 generation gives plants that are both purple and white in roughly a 3-to-1 ratio. This strongly supports what we now refer to as the gene theory, because it means that somewhere in the F1 plants there must have been some kind of information pertaining to white flowers that was not lost and was expressed later with the F2 plants. This white flowering gene must simply have been hidden or silenced in the F1 plants. We can therefore refer to purple flowers as the dominant trait and white flowers as the recessive trait. This combination of dominant and recessive traits was observed for many other aspects of the plant, and precisely the same 3-to-1 ratio was observed when examining the phenotypes in the F2 generation for smooth versus wrinkled seeds, yellow versus green seeds, green versus yellow pods, and so forth. The reason for this is that each trait corresponds to a particular gene, like a gene for flower color. But each gene has two versions, or alleles, in the genome, because the gene is found on two homologous chromosomes, one from each parent. These two alleles can be of a different genotype, meaning that the sequence of nucleotides are different, and this can lead to different phenotypes when expressed, like the two different colors that are visible on the flower. Although Mendel didn't know anything about DNA, he was still able to decipher this concept of two alleles per trait. True breeding plants must have identical alleles for a particular trait, but when he performed the hybridization, he realized that one allele was dominant, in that it was expressed while the other was recessive and stayed dormant. In this case, when purple and white are hybridized, the F1 generation is all purple, so purple must be dominant. He even realized that each gamete must have only one of these alleles, such that when each plant is fertilized, one of any of the possible combinations of alleles will result. This is the law of segregation. We now understand this is true because we have learned about meiosis and the haploid daughter cells that result. In this way, Mendel was able to explain the phenotypes in the F2 generation by rationalizing that true breeding purples have 2 purple alleles and true breeding whites have 2 white alleles. So when the sperm from one fertilizes the egg from the other, all of the resulting plants in the F1 generation must have one of each allele.
What is the structure of DNA?
The DNA has repeating subunits, and those subunits are called monomers, or nucleotides specifically. A spiral, or helix. Because DNA is double-stranded, we use the phrase double helix to describe its structure. There are 4 scientists who are credited with discovering the shape of DNA, and they are Watson, Crick, Wilkins, and Franklin. All of them received a Nobel Prize for their work, except for Rosalind Franklin. She died before the prize was given.
I am Royland M. Espartero, Bachelor of Secondary Education. Thank you for watching. Thank you for watching.